Okay, so now let's take a look at the first slice. And I had written really lightly here, cell membrane structure. Okay, so we've got 8 slices of pie. We're combining slices over here, 3 and 4 to make a really big 3 over in that direction. We don't care about that one right now though. So right now we're focusing on slice number 1, the structure of the cell membrane. What I need you folks to do is to draw 4 things here, okay, in the cell membrane. Number 1, I need you folks to show how the phospholipid bilayer is set up. So again, we've got phosphate heads and 2 lipid tails. The phosphate heads are hydrophilic. They love water. So that's why they're facing outside the cell or inside the cell. Either way, the phosphate heads are pointed in the direction of water. The lipid tails hate water. Each of these heads has 2 tails, okay? They hate water. So it's logical that these lipid tails will be facing each other, hidden away from water, okay? So we call that the phospholipid bilayer because it's made up of phosphate lipids. Bi means 2, and it's 2 layers, okay? So that's what makes up the cell membrane. So that's the first thing that you need to label is phospholipid bilayer made up of phosphate heads and lipid tails. Second thing is we have these what are called channel proteins. Basically, these are proteins that sit right here. They're embedded in the phospholipid bilayer, and they act as tunnels. They're a little more complicated than that, but basically they act as tunnels. Certain things are allowed to come into the cell. Certain things can leave the cell. And we're going to get more in depth into how these work later on in some other slices of the pie, because this slice right here is simply an introduction to cell membrane structure. So that's the second thing you need to draw and label. The third thing is carrier protein, and we'll learn more about these in another one of the slices, but basically what carrier proteins do is they allow a substance, certain substances, to come in here, bind, and then they will move that substance through the cell membrane and pretty much spit it out on the inside of the cell. They also do the reverse. They take things that are inside the cell and then move them over and spit them out this way. Okay? So that's the third thing you need to draw and label is carrier protein. In handout number 35, in OneNote, the channel protein was red. The carrier protein was blue. The fourth thing I need you to do is make a couple little squiggles here, okay? In handout number 35 in OneNote, these were orange, I believe, okay? These guys are cholesterol. And you might think, well, cholesterol is bad. Why do we have cholesterol in our cells? Well, we actually do need cholesterol embedded in the cell membrane. And what it does is it increases the flexibility of the cell membrane and allows it to wiggle and get squished. And that's why we call the cell membrane a fluid mosaic. Mosaics are made up of all different kinds of structures. A mosaic is a piece of art. And a fluid mosaic means it can move very easily. Okay? So once again, you need to sketch out the phospholipid bilayer and a channel protein that goes all the way through, and a carrier protein that goes all the way through, and put some cholesterol in here randomly. Okay? That's it for cell membrane structure. And then now you can officially title it cell membrane structure either out here or in here. Okay?